Good evening again. I'm Konstantin Weissenow from RustLab, and today I'm going to tell you about our efforts in silico structure mutation. So let's watch this quick movie clip here to show you what I mean by that. So I'm going to talk about two structure prediction systems in particular from our lab, which we developed recently, which are really, really fast. So I can produce 2D structures and now more recently also 3D structures in a matter of hundreds of milliseconds, and for longer protein sequences, ballpark of seconds. And I will also show that these structure predictions are more sensitive to small changes in the query sequence than MSA-based methods. And these two features allow us to do something like this, which is what I would call a mutation movie. So this is a movie of individual structure predictions for all the point mutations you could introduce into this particular protein, and then putting them together in sequential order and rendering them as a movie file. And this is so fast, you can do this on one GPU, almost consumer-grade GPU, in a matter of minutes. Actually, it takes longer to render the individual images with PyMol than it takes to obtain these structure predictions. So before I tell you how we do this, let me briefly tell you something about myself, since I assume most of you don't know me yet. So again, I'm Konstantin Weissenow. I have a background in computer science and then bridged off into bioinformatics and have been a PhD student with Burkhard Rost in Munich since September 2019. So I'm a very visual person, so I like all kinds of simulations, visualizations, complex systems, dynamic systems, chaotic systems. So for a while, for a couple of years, I got my hands quite literally into fluid dynamics and fluid simulation. And then for a time, I tried to grow trees inside of my computer with some mixed results. And I'm also doing all kinds of visual things, which have, in fact, nothing to do with computers. So seeing that, you're probably not surprised that when I went into bioinformatics, I very quickly got interested in this, in protein structure. Now, this has quite a lot of similarities, as you can readily see. So protein structures are fascinating. They are complex, they are dynamic, they are seemingly chaotic, but not quite, because they obey underlying rules and principles that guide their formation. And I think this is also very true for visual art, and this might be one of the reasons why I'm fascinated by them. So let's talk about protein structure prediction and a brief historical overview. So for a long time, we had something I would call a classical pipeline up to, let's say, AlphaFold 1, 2018 ballpark. So there we had a multi-stage pipeline with individual tools along the way. So we were computing from this query sequence we're interested in a multiple sequence alignment. Then from this MSA, we were explicitly computing evolutionary information with POTS models, for example, giving us evolutionary coupling scores, or as David Jones did it with covariance of the MSA. Whatever it was, we were using this explicit evolutionary representation, correlated mutations mostly, to then do the first kinds of machine learning systems in the field, going to predicted 2D information, contacts first, and then for a brief intermission periods, distance probability distributions. And then we use those as a set of constraints to go into folding to get an actual full atom 3D model. Now, things have changed quite significantly, as you know. So the trend, the big picture was that more and more of these individual steps were gobbled up by machine learning, and more specifically deep learning. And now with AlphaFold 2, we have a system that goes from MSA directly to all atom 3D model, something that was just not possible a couple of years ago. What still remained, however, until now, was this step between query sequence and multiple sequence alignment, which is still not done by these systems in a machine learning way. Now, the mere existence of AlphaFold 2 is, in fact, one of the reasons for my approach, since, well, as I said, I started my PhD in 2019. AlphaFold 2 came 2020, so this was 1 year into my PhD. And as you can imagine, as someone who tries to write a dissertation in structure prediction, and in fact, I'm the only one in our lab who specifically does that, this was quite discouraging, even though it was great scientific news. So I had to realign my approach somewhat and figure out what kind of niche I could go for instead. And I wanted to go for speed. So, and not just speed for the sake of speed, but speed with a specific use case in mind, and that is to essentially explore the space of protein folds. And for that, you need to have two things in your structure prediction system if you want to go that way towards it. So you want to be fast, that's one thing. As I mentioned, you don't want to wait like half an hour for an individual prediction. You want to sample this at high speed. But also you want to be sensitive to small changes in the query sequence. And this is not something that MSA-based predictors are particularly good at, obviously. So what I'm going to show you are the systems called EMBER-2 and EMBER-3D. And they are not going the route of explicit evolutionary information or multiple sequence alignments. Instead, we're using protein language models. Now, protein language models are a large topic by themselves, so I cannot go into detail here because of time. but essentially what they are big transformer-based architectures which are trained in a self-supervised fashion on large sequence databases. So just protein sequence, no structural information, no other protein property except just the sequence. So we're using ProtT5, and essentially what we're doing is we're throwing this query sequence into ProtT5 and then predicting, in the case of EMBER2, distances, and then go to explicit folding to get 3D models. Or more recently with EMBER3D, which is still in development, we are kind of having what I would say a true end-to-end prediction system. Since we're not computing MSAs, we're really having machine learning from the query sequence to the 3D model. So let's take a look at the systems. Actually, no, let's first talk about the history of protein language model prediction, since I started doing this quite a while ago. So during my master thesis in 2018, we had the first protein language models available. And this is kind of a timeline of the performance of those different protein language models. I measure this here in terms of contact prediction performance, since I had a 2D predictor, in terms of MCC, so higher is better. And what we wanted to beat here was our MSA-based baseline, so explicit evolutionary information. These are Weidand plots on the validation set that we've been using for all of these examples here, and the bar in the middle is, of course, the average. So with the first protein language models we had, SECVAC, back then we were absolutely not competing with evolutionary information. In fact, back then in my master thesis, I was not trying this at all. I was trying to enrich evolutionary information with protein language model embeddings instead. Now, over the years, we saw a couple of quite significant boosts in performance through better and bigger language models like ProtBERT in 2020. This is the one I competed with in CUSP-14 2 years ago. But it really took until ProtT5 from last year. This was the first time when we had a protein language model with which we could actually compete in protein structure prediction and even slightly outperform our baseline. If you're interested in ProtT5, just look at the ProtTrans paper. There's a lot of technical detail on what's going on there. So let's talk about EMBER2. This is the 2D predictor, which is published at this point. It's available on GitHub, obviously. You can look at the manuscript in structure now. How it works is we throw in the query sequence into our ProtT5 language model. And what we extract from this transformer-based architecture are specifically the attention heads, the attention matrices of this transformer. Now, this is advantageous for this approach since this is pairwise information, right? This is residue to residue information. And what EMBER2 infers is also pairwise because it infers distance probability distributions. Now, what we observed was when you extract these attention matrices from ProtT5 for a query sequence and do a little bit of post-processing, like symmetrizing it and APC, which is usually done, we get, if we visualize it, something like this. Now, this was surprising to us when we compared this with the distances or contact maps from the experimental structure from PDB for that sample. You can see, hopefully you can see it well enough, there's this faint signal here, which interestingly resembles what we see in a contact or distance map. This is surprising because this information is just the extracted embedding from the language model. And the language model, again, has not been trained on structure at all, right? This has just been trained in a self-supervised way on sequences. And yet it still picks up something that we could say is like co-evolutionary information. Now, obviously, this then motivated our approach. We were saying, okay, great, we can throw a convolutional neural network, a deep learning system on this, and predict distance maps. And this worked out quite nicely. By the way, this is a pen and ink kind of graphical representation of the approach. So I didn't want to stop there. So after publishing this, I immediately started working on EMBER3D, which was a completely new system, so not based on EMBER2. Not quite new in the sense that it is heavily based on Rosetta Fold. So at that point in time, we had a lot of information available from OpenFold, the open source implementation from AlphaFold, and Rosetta Fold, which is very accessible. It's very clean source code, so it was absolutely possible to get into this. And essentially, I took many ideas from Rosetta Fold and put them into this system. So what you see here are the main backbone parts, which are also in use in Rosetta Fold. So we're having this 2-track block here and this 3-track pipeline later in the architecture, where 1D and 2D information and then 1D, 2D, and 3D information is processed in a shared way. And then it outputs the same as Rosetta Fold, 2D features like distance maps and angle maps, and immediately outputs 3D backbone coordinates and predicted confidence values measured in LDDT. Now, what I did to Rosetta Fold was I condensed it down to the absolute bare minimum that it needs to be functional, essentially. And also did a lot of work under the hood. For example, I completely replaced the SE3 transformer implementation in the 3D part. put out a faster version from NVIDIA, which essentially doubled the speed, which was pretty good, and did a couple other smaller optimizations. So this also brought down the number of free parameters quite significantly from 130 in the original Rosetta Fold to 4.6 in our model. AlphaFold 2, I think, is in the ballpark of 200-something million parameters. So it's much leaner, it's much faster to use. And obviously, what we also did here is we are not training on MSAs or templates. All we're throwing in is Prot-5 sequence embeddings in the 1D part, where usually you have MSA-based information. And in the 2D part, we're throwing in, again, these attention matrices from the transformer instead of template information. So again, Ember3D is still a prototype. It's not yet published. We hopefully have something soon on bioRxiv in the next couple of weeks, but there is code available. So I made a pre-release. If you want to try this out, you can absolutely do so. So let's get into structure mutation. So again, our hypothesis was that our systems might be more sensitive to small changes in the underlying query sequence. Why? Because if you build an MSA, then the MSA is going to be roughly the same if you just introduce a point mutation, right? The aligned sequences are going to stay essentially the same. Maybe the order changes a little bit, but essentially it's the same MSA you get out. Now, for our predictor based on protein language models, that's not necessarily the case, because when we change something in the sequence, the embedding will change from the protein language model, and then our system might pick this up and correctly predict whatever impact on the structure this might have. Now, validating this is actually tricky because we do not have like large arrays of experimental data in PDB where we know that for a set of point mutations, the structure changes like such or so. Now, what we're doing is instead is we're validating by proxy. So we're using deep mutational scanning for this. So deep mutational scanning is an experimental technique where we introduce point mutations for, if possible, all residues in a protein sequence, and then it measures the functional impact, for example, through binding assays. Now, functional impact is not the same as structural impact, right? So you could have a small structural change, but if it's in a binding site of a protein, the small structural change might have a very big impact on the function. Conversely, you might have a relatively large structural impact in some area of the protein which is not related to the function, and so it's just happily doing whatever it was doing, and it will not impact the function. So we knew it would not have a great correlation with deep mutational scanning scores, but we were interested in whether or not we would correlate better than AlphaFold 2. And the answer to that is clearly yes, we do. So what you see here is the Spearman rank correlation of the structure delta we get between wild type and mutant, produced by Ember2, Ember3D, and AlphaFold, and the DMS scores that we have for these samples. So I'm showing this for 9 proteins in total. For 5 of them, we have AlphaFold data. For those not, and simply because of runtime. So getting these thousands of structure predictions from AlphaFold just took a long time. So we did it for the shortest sequences and not for the longer ones. And as you can see, for the 5 ones where we have AlphaFold data, for 3 of the 5, we have virtually no correlation for AlphaFold, and for the other 2 cases, a weak one, whereas Ember2 and Ember3D correlate much more strongly. Now, in terms of runtime, this is the other big argument. So AlphaFold for these 5 samples, which is about 25,000 sequences, took almost a week to compute on a single GPU, whereas Ember3D can do this in less than half an hour. And by the way, Ember2 is in the same ballpark of speed generally. So I was talking about the protein mutation movies. So this is something we can do then in a matter of minutes for small proteins. So we can produce these individual frames based on the point mutations. You see the point mutations going through up here in sequential order. You also see the currently mutated position here in blue, walking through the sequence. The other color coding indicates the confidence of the system. So whatever is more greenish is higher confidence, so a very reliable prediction. And what is more yellowish, reddish is of lower quality, or it might just be a naturally disordered region, as we have it for AlphaFold predictions as well. Synchronized to that, you have the distance map here in the output, and you have an overall mutation profile where you see which positions and which mutations have the largest impact. We're actually now getting into exactly those positions which are predicted to have a high impact for this sample. And this also makes intuitive sense since these positions here are kind of inside the protein, so it might very well be that changing something here might severely disrupt the structure of the fold. Okay, so I talked a lot about speed and sensitivity, but let's also talk about just quality of predictions really briefly, even though this is not the main focus of our approach. So if you compare this to other methods, it's pretty clear that AlphaFold obviously beats us, and this was to be expected. However, EMBER-3D is on the level of last generation structure prediction methods. So we are basically at the same average TM score as RaptorX Contact on our test sets, which are based on CUSP 13 and 14. However, disclaimer, when we're going for quality, we're applying additional relaxation steps with PyRosetta. Otherwise, the quality is a little bit lower. Now, just for a quick visual assessment, these are samples from CUSP 15, which is currently ongoing, where we see AlphaFold structures in green and EMBER-3D structures in cyan overlaid. As you can see, for many of them, we also have a pretty good agreement. So hopefully, this is also going to perform well. So for the last couple of minutes, I want to get risky and try a live demo. So I have a short, I did a brief work on a web server here, where we can throw in some sequences and get predictions from EMBER-3D. So I had to choose some sequences to throw into this, right? So I would have liked to have like a button in PDB where I could just say, give me a random sample from PDB. This would have been very cool. I don't think this exists. If it does, please let me know. Instead, what I did is I just looked at the front page today and chose some samples which were released yesterday in PDB, just to bring this point across that this was not part of the training set, okay? So let's just throw this into the web server. And as you can see, you get pretty much an instantaneous result where you see what the general idea of the structure is and also confidence values. So the more bluish it is, the more confident it is. Let's look at this one here real quick. Just want to show you a couple of examples. This is a shorter protein. So you see you get pretty much instantaneous feedback in this case. And for this one here, let's see, looks kind of like this. So there's a bit of a either disordered tail or just low prediction quality, but the core of the protein is predicted quite nicely. Now, again, the quality is not the focus here. Really, the big value of this tool is that you can almost dynamically in a live mode change the sequence. So you can just throw out a couple of amino acids and throw in a bit of tryptophan, whatever, and you get an immediate feedback on what kind of impact this has on the structure, right? Maybe we can get rid of this disordered section here anyway. Don't need that. And then at some point, we'll probably disrupt the structure significantly. Let's throw in a bit of lysine in there. So you get the idea. You can essentially work live with this protein structure. Now, obviously, this is even better if you do it in some kind of automated fashion with some tools. You can use the scripts that we provide in the repository as kind of a general guideline on how to do that. You can easily write your own scripts. We provide scripts for just regular structure prediction. We provide scripts for mutation effect prediction. We have scripts for the movie generation. So if you want to have one of these protein movies, you can do that. And we have this web server as well. By the way, this looks like a publicly available domain. It is not. You cannot reach it. We just have this internally. But we have the web server script. So if you want, you can run this on your machine. So we hope this is going to be useful for protein design, maybe for protein stability analysis, large-scale analysis of impact of mutations in general. If you use the system for something, please let us know. If you have any kind of feedback, I would be very excited to hear from you. You can find it EMBER-3D on GitHub. EMBER-2 is also available. Just let me know if you need any help. So with that, I want to close. I want to thank the organizers from 3DSIC and from ISMB in general, and in particular also my lab, my colleagues, and Burkhard for their invaluable help. Thanks a lot for having me.